the pathway of activation and regulation of hormones. As we begin our examination of what hormones really do, we must remember that regardless of the classification of the hormones, hormones by themselves really don't do anything for the cell. Instead, they start a cascade of events that lead to a change in cellular function by eventually turning the genetic machinery of the cell either on or off. It is the following cascade of events that leads to the modification and eventual adaptation of the tissues due to the initial homeostatic disruption or signals from the hypothalamus and pituitary leading to downstream modifications at the various tissues. It is the downstream modification that leads to many of the issues of hormonal responses, whether positive adaptation or a disease state, that is based on the receptor mechanism and intracellular response to the initial receptor signal. It is this signaling response that leads to a unique response within each of the cells based on the type of hormone and the metabolic action that will lead to either maintaining or returning to homeostasis that distinguishes each of the events leading to the resultant response based on the stimulus of initiation. We need to remember that it is a unique response that is specific to and leads to an adaptation based on the stressor that initiates the hormone response and not the hormone itself. More importantly, we can overlay any hormone and any adaptation attributed to that hormone into this following sequence of events. The rhythmicity and pulsatility of release of the hormones. Part of the cascade of events is dependent upon the release of hormones from the various tissues. The pattern of release from the secretory cells at the tissues is highly rhythmic, with the pattern of release being based on integrated tropic regulation from the hypothalamus and the pituitary glands, with the tissue production based on homeostatic balance. Within this, there are two distinct rhythms that are established. The two distinct rhythms that are established are based on a pulse release of the hormone, where a pulse is simply the amount of hormone released at a single time. These two pathways are either circadian, the normal cyclic release of the hormone, based on classical feedback regulation, or the ultradian pulse regulation, which is where we have a change in the normal cyclic pulse release based on an acute homeostatic disruption or various secondary signaling. Here we see the normal circadian rhythm of release, where we have a peak concentration, a minimal concentration, and the average concentration within the plasma for the hormone. This peak, this minimum, and this average is based strictly on the feedback regulation due to the concentration of the hormone in circulation. How is this pulsatility regulated? This pulsatility is regulated based on the concentration of the hormone in the plasma, where a diminishing level of concentration leads to a stimulation on the hypothalamus of the pituitary to release tropic hormones, as well as on the endocrine gland itself. This causes a release of the hormone. This release of the hormone causes a feedback inhibition on the levels above it at the endocrine gland as an autocrine, on the anterior pituitary, and on the hypothalamus, based on both the release of the tropic hormone as well as the release of the hormone itself. Once in circulation, we get interaction at the target tissues. This interaction at the target tissues causes changes in the metabolic processes of the tissues, as well as a diminishment of the hormone concentration itself. It's the balance between the high level and the low level of concentration in the plasma that dictates the normal circadian rhythmicity. But what happens when we have a stress? When we have a stress, while the average concentration will not change, the peak concentration and the minimum concentration have a huge flux. This huge flux causes a rapid release of hormone from the endocrine gland, as well as a rapid metabolism of the hormone at its target tissues. While we see greater concentrations and lower concentrations of the hormone based on this ultradian response, we do not see a greater total amount of hormone throughout the day. How is this pulsatility changed? It's the homeostatic disturbance and the metabolic stress that cause a stimulation on the hypothalamus and the pituitary. Additionally, we get a diminishment of the hormone within circulation. The stimulation on the hypothalamus and pituitary causes a release of the tropic hormone, which stimulates the endocrine gland to release the hormone into circulation. This release of hormone into circulation heads to the target tissues in an attempt to cause a metabolic change within the tissues themselves. It's the metabolic change within the tissues themselves that attempts to minimize the homeostatic disturbance and metabolic stress that initiated the ultradian response in the first place. Within the ultradian response, we will not see a reduction in hormone production until the homeostatic disturbance has gone away or the metabolic stress has gone away. Once the metabolic stresses have gone away, the ultradian response will strictly be based on that circulating level of concentration of the hormone within the plasma.
So why the difference? The difference is based on the need to modulate the responses at the tissue based on the homeostatic stress being felt by the body. As such, it is the homeostatic stress that will determine the length of change in response and the total amount of hormonal response that is necessary in order to get the body back to homeostatic balance. But because of tissue-specific adaptations that will occur, what was once seen as a stressor for the body may not be recognized as a stressor again, as the body has adapted to the initial stressor. This response is based on the idea of a specific adaptation to the imposed demands of the stressor, also known as the SAID principle. Things to think about before we move on to actions at the tissues. What do hormones interact with at the target tissues or cells? What is a pulse rate for a hormone? How does stress change the amount of hormone being produced and or released? What does it mean to have a circadian pulse rate?